All right, today we're diving into one of the biggest, most mind-blowing scientific breakthroughs of our time, CRISPR. Yeah, we're talking about the technology that's literally giving us the power to edit the code of life itself.
But let's start with a question. Our DNA is basically the instruction manual for our entire body. But sometimes that manual has typos, little mutations that can cause some really devastating diseases. So what if we could just go in, find those typos, and correct them?
So, where did this unbelievable technology come from? Well, you'd probably guess it was invented in some high-tech genetics lab, right? But the truth is, its origins are way more surprising, hidden away in the world of humble bacteria. The whole story kicks off back in the 1980s. Scientists were looking at bacterial DNA and kept seeing these weird, repeating patterns. For decades, nobody really knew what they were for. Then, fast forward to 2007, and bam, a huge breakthrough. Researchers figured out it's actually a super sophisticated immune system that bacteria use to fight off viruses. And then, just 5 years later, in 2012, scientists figured out how to hijack that natural system and turn it into the programmable gene editing tool we now know as CRISPR.
Okay, so how on earth does a bacterial defense system become a tool we can use? Like a pair of molecular scissors we can actually control. Let's get into how it works.
First off, that name, CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. I know, it's a total mouthful, but the idea behind it is actually pretty simple. Just think of it like a genetic most wanted list for viruses. Whenever a bacterium gets attacked by a virus and survives, it snips off a piece of the virus's DNA and stores it, kind of like taking a mugshot. And this all happens in 3 basic steps. Step 1 is acquisition. That's when the bacterium grabs that piece of viral DNA, our mugshot. Step 2 is biogenesis. It makes an RNA copy of that mugshot, which acts as a guide. And finally, step 3, interference. This is where that guide RNA joins forces with a protein called Cas9. Think of Cas9 as the enforcer. Together, they patrol the cell, and if they ever find DNA that matches that guide, Cas9 cuts it up, destroying the threat. Now, this is where it gets really brilliant. This is the human innovation part. Scientists realized they could create their own synthetic single-guide RNA. This means we can program it to match any DNA sequence we want. We can tell those Cas9 scissors exactly where to go and what to cut. Not just in bacteria, but in plants, animals, you name it. Even us. And this is why it was such a revolution in biology. I mean, we had gene editing tools before CRISPR, but they were so clunky and expensive. You had to build a whole new custom protein for every single gene you wanted to target. It took forever. With CRISPR, it's cheap, it's fast, and it's incredibly easy to reprogram. All you have to do is change that tiny little guide RNA sequence. It completely changed the game. But here's the thing. CRISPR isn't just one tool anymore. It's not a one-trick pony. It has evolved into a whole toolkit that can do a lot more than just cut DNA. So your standard issue Cas9 cuts right through both strands of DNA. That's perfect if you just want to turn a gene off. But scientists have cooked up other versions. There's a Cas9 nickase that only nicks one strand, which is great for making super precise repairs. And my favorite, dead Cas9. It can't cut all. Instead, it's like a programmable drone that you can send to any spot on the genome to drop off a package. Maybe something that turns a gene on, or turns it off, or even makes it glow so you can see it. And it just keeps getting better. Now we have these next-gen technologies called base editing and prime editing. These tools are even more precise. They can change single letters of DNA, literally correcting a single typo, without having to make that big cut across the DNA. It's like we've gone from using molecular scissors to using a molecular pencil and eraser. Okay, the tech is amazing, we get it. But what does it actually mean for us? What's the real-world impact that CRISPR is having right now? Well, the applications are just massive, touching almost every part of the life sciences. We're talking about gene therapies for diseases like sickle cell anemia. We're talking about brand new, super fast diagnostic tests. It's speeding up the discovery of new drugs, and it's helping us grow crops that are hardier and more nutritious. It's everywhere. So, what made CRISPR the true game changer? It really comes down to this: efficiency. The older tools, well, they were a shot in the dark. They might only work between 1 and 50% of the time. You just couldn't rely on them. CRISPR, on the other hand, can have success rates over 70%. That is a gigantic leap. It's what took gene editing from a cool science experiment to a technology that can actually change the world. Look at diagnostics, for instance. The old way, using PCR, is powerful, But you need big, expensive machines in a lab, and it takes time. But these new CRISPR-based diagnostics, with cool names like Sherlock, are a whole different story. They're so sensitive and so simple that sometimes you can get a result on a little paper strip in minutes, not hours.
And in gene therapy, this is where it gets really profound. Take a disease like sickle cell anemia. It's caused by a single typo in one gene. So the idea is just beautifully elegant. Doctors can take some of a patient's own blood stem cells, use CRISPR to fix that one typo in the lab, and then put the corrected cells back into the patient. The potential there is for a one-time permanent cure. Just incredible. But, when you have the power to literally rewrite the code of life, that comes with some enormous responsibilities. And that leads us, of course, to the massive, unavoidable ethical questions. There's a lot to think about here. You've got the technical worries, like off-target effects. What if we accidentally edit the wrong place? You've got huge social questions, like who gets access to these treatments? Will they only be for the rich? And of course, there's the fear of the unknown. What are the long-term consequences? But the biggest, most challenging issue of all is something called germline editing. And this quote really sums it up. See, editing the regular cells in a person's body only affects that one person. But editing germline cells, that means sperm, eggs, or embryos, creates changes that are passed down. They become permanent. You're not just editing one person, you're editing their children and their children's children, forever altering the human gene pool. That's a line that right now the entire global scientific community agrees we should not cross without a serious, long public conversation.
So this isn't science fiction anymore. The technology is here, right now. CRISPR has handed us the pen. We can now rewrite the book of life. And the biggest question we all have to face is, how do we decide what to write?